Hi everyone, welcome back to BiomedMariam channel. Today's video is going to be about proteins, which is the IB Biology topic 2.4. So what are proteins? Proteins are large molecules made from amino acids joined together to form chains. Amino acids are the building blocks of proteins. There are 20 different amino acids and they are linked together by peptide bonds to form a protein. The specific sequence of amino acids determines the protein's function. All amino acids have the same basic structure, a carbon atom in the center bonded to an amine group, a carboxyl group, a hydrogen atom, and an R group. The amine group in the carboxyl groups are used making the peptide bond, so does the R group that gives the polypeptide its character. The condensation reaction involves the amine group of one amino acid and the carboxyl group of another. Water is eliminated. A new bond is formed between two amino acids called a peptide bond. Protein structure can be described at several different levels. The primary structure refers to the specific sequence of amino acids. The secondary structure refers to the way the protein folds and bends, typically into alpha helices or beta sheets. The tertiary structure refers to the overall three-dimensional shape of the protein, which is determined by the interactions between the various amino acids and their side chains. The quaternary structure refers to the arrangement of multiple protein subunits within a large protein complex. Sometimes proteins can undergo denaturation, a process in which the protein structure is disrupted and the protein loses its function. This can be caused by heat, pH changes, or other physical or chemical stressors. However, some proteins can be renatured or returned to their original function by reversing the conditions that cause denaturation. The gene-to-polypeptide process, also known as protein synthesis, begins with the transcription of DNA into RNA and then the translation of that RNA into a specific sequence of amino acids. This sequence is then folded into the specific three-dimensional structure required for its function. The proteome is the complete set of proteins that is produced or modified by an organism or system, and includes all the proteins that are encoded by the genome of the organism, as well as those that are modified after transcription or translation. The proteome is dynamic and can change in response to various factors, such as environmental conditions, diseases, or cellular differentiation. Protein complexity refers to the diversity and abundance of proteins in a given organism or system. It is a measure of the number and types of proteins present, as well as their relative abundances. Protein complexity can vary significantly between different organisms, tissues, and cell types. For example, the proteome of a liver cell may be more complex than that of a red blood cell due to the diverse functions that the liver cell performs compared to the red blood cell. Protein complexity can also vary within the same organism over time, as the protein can change in response to various environmental conditions. For example, the protein of a plant leaf may change in response to different levels of light or temperature. There are many different functions of proteins in the body, including structural proteins, which provide support and structure to cells and tissues. Examples include collagen, which is a major component of connective tissue, and keratin, which is a major component of hair and nails. Hormones are proteins that are produced by endocrine glands and released into the bloodstream, where they travel to specific target cells and regulate various functions in the body. Immunity proteins, such as antibodies, protect the body against infection and disease. They recognize and neutralize foreign substances, such as bacteria and viruses. Transport proteins carry molecules and ions around the body. For example, hemoglobin carries oxygen from the lungs to the rest of the body. Sensory proteins, such as visual pigments, allow the body to perceive various stimuli, such as light and smells. Movement proteins such as actin and myosin are involved in muscle contraction and movement. Enzymes are proteins that catalyze chemical reactions in the body. They are involved in a wide range of processes, including metabolism and DNA replication. Fibrous proteins are long, thin, and string-like. They are usually insoluble in water and are found in structures such as hair and nails. Fibrous proteins are strong and flexible, which makes them well suited for their structural roles. Example of fibrous proteins is keratin, which is found in hair and nails. Globular proteins, on the other hand, are more spherical in shape. They are typically soluble in water and are found in tissues throughout the body. Globular proteins perform a wide range of functions, including catalyzing chemical reactions, transporting substances, and regulating processes. Examples of globular proteins include enzymes and hormones. One key difference between fibrous and globular proteins is their shape. Fibrous proteins are long and thin, while globular proteins are more spherical. This difference in shape is due to the way the proteins are folded, which is determined by the sequence of amino acids that make up the protein. Another difference between fibrous and globular proteins is their solubility. Fibrous proteins are generally insoluble in water, while globular proteins are soluble. This difference in solubility is due to the presence or absence of hydrophobic amino acids in the protein. Proteins that contain a high proportion of hydrophobic amino acids tend to be insoluble, while proteins that contain mostly hydrophilic amino acids tend to be soluble. Thank you for your attention. That was all we had for you today. I hope you enjoyed it. Don't forget to subscribe and like this video.